Is school an instrument of imbibing and promoting democracy? Children, they have to be nurtured into a democratic citizens. Is school an instrument on this? If so, how? I'll stop it there.
ڈیئر لرنرس، آج آپ کے کورس BUDC 104 کے ایک اہم شاعر کے حوالے سے ہم گفتگو کریں گے۔ اس گفتگو سے قبل یہ ضروری ہے کہ اس کا بیک گراؤنڈ ہم جان لیں کہ شاد عظیم آبادی کا بیک گراؤنڈ کیا ہے، اس وقت اردو غزل کس نوعیت جو ہے صورتحال میں تھی، اور اس وقت اردو غزل کے کیا تصورات لوگوں میں عام تھے۔ تو یہ تو آپ جانتے ہیں کہ کلاسیکی غزل کے بعد ہمارے اردو شاعری میں نو کلاسیکی شاعری کا آغاز ہوتا ہے۔ شاد عظیم آبادی نو کلاسیکی شعراء میں شمار کیے جاتے ہیں۔ اس وقت انگریزوں کے تسلط کے زیر اثر انگریزی شاعری کا اثر اردو پر بھی مسلط ہوا۔ اس تصورات اور اس رجحانات کے پیش نظر اردو غزل کی مخالفت کی گئی اور اردو نظم نگاری کی تحریک کو آگے بڑھانے کی کوشش کی گئی۔ آپ جانتے ہیں کہ وہ دور تھا 19ویں صدی کا آخری دور، یعنی 1857 کے بعد جب غدر ہوتا ہے اور انگریز پورے بھارت میں مسلط ہو جاتے ہیں، تو اس وقت ان کی تھنکنگ، ان کے آئیڈیالوجی یا ان کے جتنے بھی رجحانات، اعمال و افکار تھے، سب کا اثر یہاں پر پڑھنا شروع ہوا۔ تو اردو ادب بھی اس سے متاثر ہوا۔ اس تاثر میں لٹریری لحاظ سے جب ہم دیکھیں گے تو غزل کی خوب مخالفت ہوئی۔ مولانا الطاف حسین حالی نے باضابطہ طور پر مقدمہ شعر و شاعری لکھی، اور مقدمہ شعر و شاعری لکھ کر مثنوی، مرثیہ کی خوب تعریف کی اور قصیدہ اور غزل کی مخالفت کی، اور بلینک ورس کی سے مثالیں پیش کی اور کہا کہ اس میں منظم خیالات کو پیش نہیں کیا جا سکتا ہے۔ تو یہ سب اثر تھا انگریزی شاعری کے اثرات اردو شاعری پر مرتب ہونا شروع ہوئے۔ اس دور میں جس وقت غزل ڈگمگا رہی تھی، اس وقت شاد عظیم آبادی نے غزل کو بہت ہی سمجھداری سے تھامے رکھا اور غزل گوئی میں نئے امکانات پیدا کیے۔ تو آئیے سب سے پہلے ہم غور کرتے ہیں کہ شاد عظیم آبادی نے غزل میں تنوع کیسے پیدا کیا۔ پہلے تو شاد عظیم آبادی کا کچھ تعارف ہو جائے، تو اس کے لیے جہاں سلائیڈ کی طرف آپ دیکھیں گے، یعنی جہاں پہ غزل کا تعارف شروع ہوتا ہے، تو آپ سب سے پہلے یہ جاننے کی کوشش کریے کہ علی محمد شاد عظیم آبادی کا تعلق ایک خوشحال اور اعلیٰ علمی خاندان سے تھا۔ پٹنہ میں 8 جنوری 1846 کو ان کی پیدائش ہوئی۔ اس زمانے کے رواج کے مطابق 4 سال کی عمر میں ابتدائی تعلیم کا سلسلہ انہوں نے گھر پر ہی شروع کیا۔ ابتدا میں انہوں نے عربی اور فارسی، بعد میں اردو کی تعلیم حاصل کی۔ اس کے بعد انہوں نے انگریزی زبان بھی سیکھی۔ لائق اساتذہ کی تربیت اور ذاتی لیاقت نے ان کے علمی ذوق کو جلا بخشی، چنانچہ انہوں نے بہت جلد عربی، فارسی اور اردو زبانوں میں مہارت حاصل کر لی۔ مذہبی علوم سے بھی انہیں خاص دلچسپی تھی۔ انہوں نے اسلامی علوم کے ساتھ ساتھ دیگر مذاہب، عیسائی مذہب، پارسی مذہب، ہندو مذہب اور بھی دیگر مذاہب کا انہوں نے مطالعہ کیا۔ اس طرح ان کا مطالعہ بہت وسیع تر ہو گیا۔ دیکھیے سامعین، اب ذرا توجہ کیجئے گا کہ جب مختلف علوم کا ہم جائزہ لیتے ہیں اور مختلف علوم کے یہ مذاہب کے لٹریچر کو پڑھتے ہیں تو ذہن میں وسعت پیدا ہوتی ہے، خیالات میں نکھار پیدا ہوتا ہے۔ تو شاد عظیم آبادی کے ساتھ بھی یہی ہوا، اس طرح ان کے فکر و نظر میں بہت ہی زیادہ وسعت بھی پیدا ہوئی اور لچک بھی پیدا ہوئی۔ یہی وجہ ہے کہ ان کے کلام میں انسانیت کا ہماگیر تصور دیکھنے کو ملتا ہے۔ انہوں نے شاعری کی ابتدا 1860 میں کی۔ شعرا کی شعر گوئی، یہ تو حقیقت ہے کہ ان کا پیدائشی ملکہ تھا، یعنی ودیت ہوئی تھی شاعری میں انہیں الفت حسین فریاد عظیم آبادی سے تلمذ حاصل ہوا۔ سب سے پہلے انہوں نے باضابطہ طور پر الفت حسین فریاد عظیم آبادی سے یہ جو کہ شاگردی میں علم کو حاصل کیا، یعنی ان کی شاعری کی اصلاح لینا شروع کیا۔ کبھی کبھی سفیر بلگرامی سے بھی انہوں نے اپنی شاعری کی اصلاح لی۔ اس کے علاوہ عبرتی اور زخمی سے بھی اصلاح لینے کا ذکر ملتا ہے کتابوں میں۔ شاد عظیم آبادی کونا مشق اور قادل الکرام شاعر ہونے کے ساتھ ساتھ ایک اچھے مصنف بھی تھے۔ نثر و نظم دونوں میں انہوں نے کئی کتابیں یادگار چھوڑیں۔ شاعری میں ان سے 8 مثنویاں، نالہ فریاد، دلکش، نغمہ جاں فضا، یاد حبیب، چشمہ کوثر، ثمرہ زندگی، نشان دلکش اور نوید ہند منسوب ہیں۔ ایک اور مثنوی مادر وطن بھی ان کی یادگار ہے۔ فکر بلیغ ان کے مرثیوں کا مجموعہ ہے۔ ظہور رحمت ان کی رباعیات کا مجموعہ ہے۔ اب ذرا دیکھیے کہ یہ کتابیں جب ہم دیکھتے ہیں تو اس سے یہ اندازہ ہوا کہ شاد عظیم آبادی نے مختلف اصناف سخن پر طبع آزمائی کی۔ ان کے یہاں باضابطہ طور پہ مثنوی بھی ہے، مرثیہ بھی ہے، رباعی بھی ہے، قطعات بھی ہے، اور اس کے علاوہ دیگر اصناف میں بھی انہوں نے طبع آزمائی کی۔ لیکن ان کی جو شناخت بنتی ہے یعنی ادبی دنیا میں جو ان کی پہچان بنتی ہے وہ اردو غزل سے بنتی ہے۔ جیسے ابھی مثال دیا تھا فکر بلیغ، یہ ان کے مرثیوں کا مجموعہ ہے۔ اب اس میں انہوں نے ایک مقدمہ بھی لکھا ہے جس پہ انہوں نے بہت معنی خیز گفتگو کی ہے۔ تو شاد عظیم آبادی کی نثری تصانیف کا بھی ہم ذکر کرتے ہوئے آگے بڑھتے ہیں۔ اب دیکھیے سلائیڈ کی طرف ذرا دیکھیے کہ شاد عظیم آبادی نے غزل کے ساتھ ساتھ جو ہیں وہ مرثیہ، مثنوی، نظم، قطعات اور رباعیات میں بھی طبع آزمائی کی، لیکن ان کا اصل جوہر ان کی غزل گوئی میں ظاہر ہوا اور یہی ان کی شہرت کا سبب بھی بنا۔ صنف غزل سے ان کے مزاج کو ایک خاص مناسبت یعنی لگاؤ تھا۔ اپنی غزلوں میں انہوں نے روایت کی پاسداری بھی کی ہے، اسے جدید خیالات، نئے موضوعات اور اسالیب سے ہم آہنگ بھی کیا ہے۔ انہوں نے جب شاعری کے میدان میں قدم رکھا تو نظم اور نظم جدید کا دورہ تھا۔ زیادہ تر یہ ان کا معاملہ جو تھا وہ اس وقت نظم کی مخالفت ہو رہی تھی۔ اس سے قبل والے حصے میں میں نے آپ کو بیان کیا تھا جہاں تعارف کیا کہ انہوں نے باضابطہ طور پہ سروع سے ہشتی جو ان کے قطعات کا مجموعہ ہے وہ بھی انہوں نے لکھا۔ شاد عظیم آبادی کی نثری تصانیف بھی بہت اہمیت کی حامل ہیں۔ مثلا حیات فریاد ان کے استاد الفت حسین فریاد کے حالات زندگی اور کلام کے تبصرے پر مشتمل تھا۔ مکتوبات شاد ان کے خطوط کا مجموعہ ہے۔ اسی طرح تاریخ بہار، شان ریاست، صورت الخیال، مردم دیدہ، شاد کی کہانی، شاد کی زبانی، نقش پائیدار اور نوائے وطن وغیرہ ان کی اہم تصانیف ہیں۔ شاد عظیم آبادی کی مجموعی علمی و ادبی خدمات کے اعتراف میں حکومت برطانیہ نے انہیں 1891 میں خان بہادر کا خطاب عطا کیا اور 1000 روپیہ سالانہ وظیفہ بھی مقرر کیا۔ ان کا انتقال 7 جنوری 1927 میں ہوا۔ یہ مختصر تعارف آپ کے سامنے ابھی جو میں نے پیش کیا۔ اب اس میں دیکھیے کہ ان کی زندگی میں، ان کے فکر سخن میں ڈائمنشن ہیں، جہتیں ہیں، اور جب کوئی شاعر مختلف جہتوں کے لحاظ سے کام کرتا ہے اور مختلف جہتوں پر کمانڈ رکھتا ہے تو اس کی شاعری میں وسعت پیدا ہو جاتی ہے، گہرائی پیدا ہو جاتی ہے۔ ابھی جیسا کہ میں ذکر کر رہا تھا کہ وہ دور جو تھا اس دور میں غزل کی مخالفت ہو رہی تھی، بعض لوگوں نے یہ کہا کہ غزل معشوق کی موہوم کمر ہے، ایک رجحان بنا دیا گیا غزل کے خلاف۔ لیکن اس دور میں بھی شاد عظیم آبادی نے یہ ثابت کیا کہ اردو غزل ایک ایسا فارم ہے، ایک ایسی صنف ہے جس میں ہر طرح کے موضوعات، ہر طرح کے مسائل، ہر طرح کے معاملات اس میں پیش کیے جائیں گے۔ یہ شاد عظیم آبادی کا بہت بڑا کمال تھا، جنہوں نے اس دور میں، جس دور میں غزل کی تحقیر کی جا رہی تھی، تذلیل کی جا رہی تھی، یعنی اس کو کمتر دکھانے کی کوشش ہو رہی تھی، اس وقت شاد عظیم آبادی جیسے نو کلاسیکی شعراء غزل کی روایت کو آگے بڑھانے میں منہمک تھے۔ اب آئیے ان کی غزل گوئی کے کچھ بنیادی نکات کی طرف ہم گفتگو کرتے ہیں۔ تو اس سے پہلے اب سلائیڈ کی طرف ہم جب بڑھیں گے تو یہ تو آپ کو معلوم ہے، جیسا کہ ابھی میں نے عرض کیا تھا، کہ شاد عظیم آبادی مختلف اصناف کے یہ پر تبازمائی کی، اور مختلف اصناف پر تبازمائی کرنے کے ساتھ ساتھ انہوں نے نثر پر بھی یہ جو ہے اچھا خاصا سرمایہ انہوں نے پیش کیا۔ لیکن ان کی اصلی پہچان جو بنتی ہے، ان کی ایک جو شناخت بنتی ہے، جو ان کا جوہر کھلتا ہے، وہ غزل سے ظاہر ہوتا ہے۔ جدید خیالات، نئے موضوعات اور اسالیب سے انہوں نے اپنی غزلوں کو ہم آہنگ کیا۔ انہوں نے جب شاعری کے میدان میں قدم رکھا تو نظم اور نظم جدید کا باضابطہ طور پہ رجحان غالب تھا۔ زیادہ تر شعراء قومی، ملکی، تہذیبی، سماجی موضوعات پر نظمیں لکھ رہے تھے، لیکن اس کے باوجود وہ اپنے کلام میں حسن و عشق کی مختلف کیفیات، واردات کی ترجمانی سے بے نیاز نہیں تھے، بلکہ غزل کی مقبولیت کے اسباب میں شاد عظیم آبادی منہمک تھے۔ اب ہم آگے چلتے ہیں، تو اس سے پہلے ایک بات میں یہ عرض کرنا چاہتا ہوں۔ اب میری بات کرنا یہ جو ہے توجہ دیجئے، سلائیڈ پہ نظر مت رکھیں۔ میں یہ کہنا چاہتا ہوں کہ وہاں پر جو ہے باضابطہ طور پر جن موضوعات کو روایت کے طور پر غزل میں برتا جا رہا تھا، وہ ایک محدود دائرہ تھا، یعنی حسن و عشق، اور لوگوں نے اسی لیے اس کی مخالفت بھی کی۔ جدید علوم جب آیا مغرب سے تو اس کے زیر اثر لوگوں نے دیکھا کہ ارے جدید علوم میں تو مختلف زندگی گزارنے کے مختلف نئے طریقے موجود ہیں، جدید علوم کے مختلف جہتیں موجود ہیں، اور ہم وہی اپنی پرانی روایت پہ چلے آ رہے ہیں، وہی پرانی ریت رواج کے پابند ہوتے جا رہے ہیں۔ تو یہ بنیادی یہ جو ہے یہ غزل پہ اعتراض تھا۔ یہ حقیقت ہے کہ غزل کا بنیادی محور اور بنیادی مرکز جو ہے وہ باضابطہ طور پر حسن و عشق ہے، لیکن حسن و عشق ایک سمبل ہوا کرتا تھا۔ کیوں؟ اس لیے کہ عشق کے جذبے کے بغیر کوئی انسان اس دنیا میں زندہ نہیں رہ سکتا ہے۔ اگر اس کے دل میں جذبہ نہ ہو، اگر وہ کیفیت نہ ہو عشق کی، تو پھر وہ زندہ نہیں رہ سکتا ہے۔ تو عشق کے پردے میں انسانی زندگی کو گزارنے کا سلیقہ سکھلایا جاتا تھا۔ آپ کو معلوم ہوگا میر تقی میر نے اپنے میر تقی میر کے والد نے میر سے کہا کہ بیٹا عشق اختیار کرو۔ اسی طریقے سے ہماری روایت میں عشق کی بہت اہمیت ہے۔ تو جو غزل میں عشق کا تصور ہے، حسن و عاشقی کا جو تصور ہے، وہ دراصل جو ہے انسانی عظمت کا بیان ہے۔ وہ حسن و عشق کے پردے میں انسان کو سمجھانے کا ایک پلیٹ فارم ہے، اس لیے اردو غزل میں عشق و عاشقی کو خوب برتا گیا ہے۔ اب آئیے، اب دیکھیے سلائیڈ کی جب آپ دیکھیں گے تو خالص عشقیہ شاعری کے جملہ پہلوؤں کی عکاسی غزل میں ہو رہی تھی، پوری آب و تاب سے نمایاں ہو رہی تھی۔ یہ بالکل حقیقت ہے۔ غزل زندگی کے اس رشتے کو گلے لگانے اور نبھانے میں زیادہ وفادار ثابت ہوئی۔ یہ بھی حقیقت ہے کہ زیادہ تر موضوعات اور مضامین عشق و عاشقی کے تھے۔ اس نے نئے پیرائے میں حسن و عشق کی ترجمانی کے ساتھ زندگی کو آئینہ دکھانے کی بھی ذمہ داری قبول کی۔ یہ صنف غزل کی بنیادی خصوصیت تھی کہ وہ تازہ دم ہو گئی، یعنی اپ ڈیٹ ہو گئی، اور اس نے کیا حسن و عشق کی ترجمانی کے ساتھ ساتھ زندگی کے تقاضے کو بھی وہ اپنے اندر سمونا شروع کر دیا۔ شاد کی غزلوں میں اس کیفیت کو دیکھا اور محسوس کیا۔ جب شاد نے یہ دیکھا کہ یہ ہو رہا ہے تو ان کی غزلوں میں انہوں نے یہ اپنی غزلوں میں ان تمام چیزوں کو شامل کر لیا۔ انہوں نے احساس دلایا کہ غزل صرف گل و بلبل اور حسن و عشق کی داستان سرائی کا نام نہیں، یعنی غزل صرف گل و بلبل اور حسن و عشق کی داستان سرائی کا نہیں بلکہ ایک لطیف انسانی جذبے کے اظہار کا نام ہے۔ اس کی مختلف صورتیں ہو سکتی ہیں اور ہر صورت اپنی جگہ خاص اہمیت رکھتی ہے۔ ان کی انفرادیت یہ ہے، یعنی شاد عظیم آبادی کی جو سب سے منفرد کا نام پہچان ہے وہ یہ ہے کہ انہوں نے حضرت موہانی سے بہت پہلے غزل کی احیا کی طرف اس وقت توجہ دی جب غزل بہت بدنام اور ہر طرف سے نرغے میں تھی۔ انہوں نے روایت کی پاسداری کرتے ہوئے غزل کو نئے موضوعات اور نئے احساسات سے ہمکنار کیا۔ اس طرح ان کی شناخت جدید دور کے اہم غزل گو شاعر کی حیثیت سے ہوتی ہے۔ سامعین و ناظرین، شاد عظیم آبادی نے حسن و عشق کی مختلف کیفیتوں کے اظہار میں سادگی، متانت دونوں کو برقرار رکھا۔ کسی طرح کی شوخی یا غیر شائستگی کو راہ نہیں دی، یعنی اس کو داخل ہونے نہیں دیا۔ زندگی کی تلخ حقیقتوں کے باوجود وہ اس بات کا اعتراف کرتے ہیں کہ کائنات کی لطیف ترین جو خوبصورتی ہے وہ عورت ہی ہے، اور عورت جن سے یہ جو ہے جن سے لطیف ہے، جس کی طرف ہر شخص جو ہے وہ ایک عزت اور اعلیٰ مقام کے تحت عورت کے مقام کا تعین کرتا ہے۔ یہی حال ہمارے ہاں غزل میں بھی ہوا کہ باضابطہ طور پہ جن سے لطیف کی رعنائی و رنگینی کو خوب پیش کیا گیا۔ بعد میں یہ جذبہ شاد عظیم آبادی کے یہاں بھی ہلکا ہو جاتا ہے، یعنی مدھم ہو جاتا ہے، صوفیانہ خیالات میں تبدیل ہو جاتا ہے اور بالآخر عشق حقیقی کا فیضان نظر آتا ہے۔ ان سب کے باوجود ان کے کلام میں حسن انسانی کی عظمت کی جادوگری کسی طرح کم ہونے کا نام نہیں لیتی ہے۔ اب جب توجہ کیجئے، اب سلائیڈ کی طرف آپ توجہ مت دیجئے بلکہ میری گفتگو پہ توجہ دیجئے۔ ذرا دیکھیے کہ انسان جو بچپن میں ہوتا ہے، پھر بچپن کے بعد جب وہ شباب کی طرف بڑھتا ہے، پھر جوان ہوتا ہے، جوانی کے بعد پھر وہ ایک کہ یہ ایسی ایج میں پہنچتا ہے جس وقت اس کا شعور بالیدہ ہو جاتا ہے، تو انسان کے یہاں باضابطہ طور پہ ارتقا ہے۔ ارتقا کسے کہتے ہیں؟ ارتقا کہتے ہیں بتدریج دھیرے 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 اوپر کو اٹھنا۔ یہی معاملہ شاعری میں بھی ہوتا ہے کہ آدمی شروع میں اپنی شاعری کا آغاز عشق و عاشقی سے شروع کرتا ہے اور پھر رفتہ رفتہ انسانی معاملات، اخلاقی اقدار، سماجی کردار اور مختلف تصوفانہ اور یہ فلسفیانہ مضامین کو پیش کرتا رہتا ہے۔ یہی معاملہ شاد عظیم آبادی کے یہاں بھی آیا کہ شاد عظیم آبادی کی شاعری کی ابتدا جو ہوتی ہے وہ تو عشق و عاشقی سے ہوتی ہے، لیکن دھیرے دھیرے وہ عصری تقاضوں کی بنیاد پر جو اس وقت کے چیلنجز تھے اس کی روشنی میں انہوں نے باضابطہ طور پہ غزل کو بہت ہی زیادہ وسعت بخشی باعتبار موضوع بھی اور باعتبار فکر بھی۔ تو ان کے یہاں اصلاً اخلاقی اقدار، انسانی اقدار بہت ہی زیادہ ان کی شاعری میں غالب ہیں۔ یہ بات ذہن میں رکھنا چاہیے کہ شاد عظیم آبادی جو ہیں وہ اظہار تعصب کے شاعر نہیں ہیں، ان کے یہاں غم نہیں ہیں، بلکہ ان کے یہاں نشاتیہ پہلو زیادہ ملتا ہے۔ ان کے یہاں انسانی اخلاق، انسانی اقدار کی ترجمانی زیادہ ملتی ہے۔ اب دیکھیے ایک شعر دیکھتے ہیں۔ ڈھونڈو گے اگر ملکو ملکو ملنے کے نہیں نایاب ہیں ہم۔ ڈھونڈو گے اگر ملکو ملنے کے نہیں نایاب ہیں ہم۔ تعبیر ہے جس کی حسرت و غم، اے ہم نفسو وہ خواب ہیں ہم۔ دیکھا آپ نے؟ اب جب سلائیڈ کی طرف توجہ دیجئے۔ ڈھونڈو گے اگر ملکو ملنے کے نہیں نایاب ہیں ہم۔ اب دیکھیے شاعر کس تصور کو بیان کر رہا ہے اس میں۔ ڈھونڈو گے اگر ملکو ملنے کے نہیں نایاب ہیں ہم، تعبیر ہے جس کی حسرت و غم، اے ہم نفسو وہ خواب ہیں ہم۔ اگلا شعر دیکھیے۔ لاکھوں ہی مسافر چلتے ہیں منزل پہ پہنچتے ہیں دو ایک۔ لاکھوں ہی مسافر چلتے ہیں منزل پہ پہنچتے ہیں دو ایک۔ اے اہل زمانہ قدر کرو نایاب نہیں کامیاب ہیں ہم۔ اے اہل زمانہ قدر کرو نایاب نہیں کامیاب ہیں ہم۔ اب یہاں جو ایک میسج دیا جا رہا ہے ابھی ہم ایکسپلین کریں گے، اس سے پہلے تیسرا شعر بھی سنیے۔ مرغان قفس کو پھولوں نے اے شاد یہ کہلا بھیجا ہے۔ مرغان قفس کو پھولوں نے اے شاد یہ کہلا بھیجا ہے، آ جاؤ جو تم کو آنا ہو، ایسے میں ابھی شاداب ہیں ہم۔ مرغان قفس کو پھولوں نے اے شاد یہ کہلا بھیجا ہے، آ جاؤ جو تم کو آنا ہے، ایسے میں ابھی شاداب ہیں ہم۔ اب توجہ دیجیے، 3 شعر پر جو ایکسپلین سنیے، سلائڈ پہ دھیان مت دیجیے۔ اب دیکھیے اس میں 3 اشعار میں 3 مختلف تصورات کو انہوں نے مختلف زاویے سے پیش کیا ہے، اور یہ سارے کے سارے جو تصورات ہیں وہ انسانی اخلاق و اقدار کے ہیں۔ ڈھونڈو گے اگر ملکو ملنے کے نہیں نایاب ہیں ہم۔ سب سے پہلے انسان کی عظمت کو بیان کیا گیا ہے کہ شخصیت کا جو ظہور ہوتا ہے، یعنی شخصیت جو بنتی ہے وہ اپنے عمل کی بنیاد سے بنتی ہے، اور اچانک کتاب کے ورق کی طرح نہیں بنتی ہے، بلکہ ایک محنت اور طویل عرصے کے بعد شخصیت ابھر کر سامنے آتی ہے۔ علامہ اقبال نے اسی کو کہا تھا، ہزاروں سال نرگس اپنی بے نوری پہ روتی ہے، بڑی مشکل سے ہوتا ہے چمن میں دیدہ ور پیدا۔ اب اسی تناظر میں اس شعر کو دیکھیے، ڈھونڈو گے اگر ملکو ملنے کے نہیں نایاب ہیں ہم، تعبیر ہے جس کی حسرت و غم، اے ہم نفسو وہ خواب ہیں ہم۔ تو شاد عظیم آبادی کی یہ بنیادی خصوصیت ہے کہ انہوں نے اخلاقی انسانی اقدار کو بہت منظم طریقے سے پیش کیا ہے، اور بہت احسن طریقے سے پیش کیا ہے، اور نشاتیہ لہجے میں بیان کیا ہے۔ دوسرا شعر جو اس میں شامل تھا، ابھی آپ نے جس کو سنا، لاکھوں ہی مسافر چلتے ہیں منزل پہ پہنچتے ہیں 2-1۔ یہ کیا ہے؟ کسی منزل پر پہنچنے کے لیے 10 آدمی چلے، اس میں سے 2 یا 1 کامیاب ہو گئے اور باقی ناکام ہو گئے۔ اب دیکھیے یہاں پہ شاد عظیم آبادی کس لفظیات میں، کس غزل کی شعریات میں لوگوں کو انسانی اقدار یعنی انسانی کارکردگی اور اس کے تحرک کی مثال پیش کرتے ہیں کہ دیکھو انسان اپنی منزل پر اسی وقت پہنچتا ہے جب اس کے اندر 3 صفات موجود ہوں۔ نمبر 1، اس کے اندر سچائی کا جذبہ ہو، سچائی ہو، ایمانداری ہو، اور تیسری چیز یہ ہے کہ محنت اور مشقت کرنا جانتا ہو۔ اگر انسان یہ 3 صفت سے متصف ہو جائے، 3 خوبیاں اپنے اندر پیدا کر لے، تو پھر وہ اپنے مشن تک پہنچ جاتا ہے۔ مشن تک پہنچنے کی یہ لازم ہے کہ اس کے اندر صداقت ہو، جذبہ ہو اور محنت ہو۔ اگر یہ تینوں خوبیاں جمع ہو جائیں تو انسان پہنچ جاتا ہے۔ اسی کو بہت عمدہ انداز میں اور بطور تمثیل کے جس اس میں پہنچے کا نام پیش کیا ہے، وہ شاعری کی کلام زبان میں نہایت ہی خوبصورت ہے۔ لاکھوں ہی مسافر چلتے ہیں منزل پہ پہنچتے ہیں 2-1، اے اہل زمانہ قدر کرو نایاب نہیں کمیاب ہیں ہم۔ مرغان قفس کو پھولوں نے اے شاد یہ کہلا بھیجا ہے، آ جاؤ جو تم کو آنا ہو، ایسے میں ابھی شاداب ہیں ہم۔ آپ نے دیکھا ہوگا کہ جب آدمی پریشان حال ہوتا ہے تو اپنے بھی اگنور کرنے لگتے ہیں، نظر انداز کرنے لگتے ہیں۔ شاعر کہہ رہا ہے کہ یہ دنیا کا دستور ہے کہ جب آپ خوشحالی کی زندگی میں ہوں گے اور جب آپ ایسو پرشتی میں ہوں گے، تمام ضروریات آپ کی پوری ہو رہی ہوں گی، تو سارے لوگ آپ کے ہوں گے۔ اگر آپ مشکل مراحل میں آ جائیں، مشکل مراحل میں جب آپ آ جاتے ہیں تو بہت سے لوگ ساتھ چھوڑ کر چلے جاتے ہیں۔ اس کے برخلاف وہ تصور یہ پیش کر رہے ہیں کہ اے دنیا والو، ابھی تمہارے پاس موقع ہے کہ تم آ جاؤ، میں ابھی ایسی حالت میں شاداب ہوں، خوشحال ہوں، اور باضابطہ طور پہ یہ جو ہے آرام و عیش میں ہوں۔ کیوں؟ اس لیے کہ مرغان قفس، آپ نے دیکھا ہوگا لفظ استعمال کیا ہے مرغان قفس۔ مرغ کے معنی چڑیا کے ہوتے ہیں، قفس کے معنی ہوتے ہیں پنجڑے کے، یعنی وہ پرندہ جو پنجڑے میں ہو۔ اب اس کا مفہوم سمجھ، پھر سنیے شعر، مرغان قفس کو پھولوں نے اے شاد یہ کہلا بھیجا ہے، یعنی ان پرندوں کو یہ پیغام بھیجا گیا ہے جو پنجڑے میں قید ہیں۔ کس نے بھیجا ہے؟ پھولوں نے بھیجا ہے۔ کیا بھیجا ہے؟ آ جاؤ جو تم کو آنا ہو، ایسے میں ابھی شاداب ہیں ہم، یعنی وقت رہتے رہتے جو کام کر لیا جاتا ہے تو وہ کام منزل تک پہنچ جاتا ہے۔ اگر وقت نکل جاتا ہے اور آدمی تساہل میں اور غفلت میں رہتا ہے تو پھر وقت ہاتھ سے جب چلا جاتا ہے تو پھر وہ کام پورا نہیں ہوتا، یعنی انسان منزل تک نہیں پہنچ سکتا ہے۔ اب آئیے اب سلائڈ کی طرف آگے بڑھتے ہیں۔ اب ذرا آپ دیکھیے سلائڈ کی طرف، شاد عظیم آبادی کی یہ، اب سلائڈ کو دیکھیے ذرا آپ۔ ہاں، شاد عظیم آبادی کی غزلوں کی جو نمایاں خصوصیات ہے، جو ابھی شعر آپ نے سنے، اس میں نرم لہجہ، بیان کی سادگی اور صفائی، سوز و گداز، کیف و سرور، اثر و تاثیر وغیرہ یہ ان کی بنیادی خصوصیات ہیں، جسے انہوں نے دلکش اور مترم انداز میں اپنی غزلوں میں پیش کیا ہے۔ ان کے کلام میں جو سنجیدگی، متانت اور معنویت پائی جاتی ہے وہ ان کے ہم عصروں بالخصوص امیر مینائی، داغ دہلوی اور جلال وغیرہ کے ہاں نظر نہیں آتی۔ ان کے کلام میں تغزل اپنی تمام تر کیفیات کے ساتھ نمایاں ہے، زبان فطری اور عام فہم ہے۔ ہندی لفظوں کا ان کے ہاں خوب استعمال بھی دیکھنے کو ملتا ہے۔ بیشتر غزلیں مترنم بحر میں ہیں اور رواں ہیں۔ محاورات، روزمرہ اور سہل ممتنع کی بھی اچھی مثالیں ملتی ہیں۔ ان کے محبوب موضوعات میں معاملات حسن و عشق کے علاوہ اخلاق، فلسفہ، تصوف اور توحید وغیرہ شامل ہیں۔ انہوں نے اپنی غزلوں میں جذبات، افکار اور اظہار کے تنوع پر زور دیا ہے اور اپنے پرلطف انداز بیان سے اسے دلچسپ بنانے کی کوشش کی ہے۔ اب دیکھیے اس ضمن میں بھی ایک شعر دیکھیے، نگہ کی برچھیاں جو سہ سکے سینہ اسی کا ہے، نگہ کی برچھیاں جو سہ سکے سینہ اسی کا ہے، ہمارا آپ کا جینا نہیں جینا اسی کا ہے، نگہ کی برچھیاں جو سہ سکے سینہ اسی کا ہے، ہمارا آپ کا جینا نہیں جینا اسی کا ہے۔ شاد عظیم آبادی کے کلام کی ایک خوبی یہ بھی ہے کہ ان کے بہت سے اشعار ضرب المثل بن گئے ہیں اور لوگوں میں یہ خاص اور عام لوگوں میں زبان زد ہو گئے ہیں وہ اشعار۔ مثلا تمناؤں میں الجھایا گیا ہوں، تمناؤں میں الجھایا گیا ہوں، کھلونے دے کے بہلایا گیا ہوں، یہ بزم میں ہے یہاں کوتاہ دستی میں ہے محرومی، جو بڑھ کر خود اٹھا لے ہاتھ میں مینا اسی کا ہے، تڑپ اے دل تڑپنے سے ذرا تسکین ہوتی ہے، خاموشی سے مصیبت اور بھی سنگین ہوتی ہے، تڑپ اے دل تڑپنے سے ذرا تسکین ہوتی ہے، خاموشی سے مصیبت اور بھی سنگین ہوتی ہے، کہاں سے لاؤں صبر حضرت ایوب اے ساقی، کہاں سے لاؤں صبر حضرت ایوب اے ساقی، خم آئے گا صراحی آئے گی تب جام آئے گا۔ شاد کا دور اگرچہ پرآشوب اور انتشار کا زمانہ تھا، لیکن ان کے کلام میں اس کی آہٹ بہت کم محسوس ہوتی ہے۔ انہوں نے رنج و علم اور ناکامی و محرومی کو اپنا شیوا نہیں بنایا، بلکہ اسے ایک ایسے وسیع تناظر میں دیکھنے کی کوشش کی جو ان کی وسیع القلبی اور آفاقی فکر کو ظاہر کرتی ہے۔ وہ حوصلہ شکنی کے نہیں بلکہ حوصلہ افزائی کے پیامبر تھے، وہ حوصلہ افزائی کے پیامبر تھے۔ چنانچہ ان کے کلام میں دردمندی، خلوص، عاجزی، صبر و رضا وغیرہ کی مثالیں کثرت سے ملتی ہیں، اور یہی وجہ ہے کہ نیاز فتح پوری، سائل دہلوی، حضرت محانی، سفیل بلگرامی، ضامن، محشر، اقبال، وحشت، مجنو گورکھپوری، فراق گورکھپوری وغیرہ نے غزل کی حیات نو بخشنے میں شاد کی صلاحیتوں کا کھلے دل سے اعتراف کیا ہے۔ شاد کو اپنے فن اور شاعری میں باضابطہ طور پر یہ بات سمجھنا چاہیے کہ شاد عظیم آبادی نے مختلف فکروں کو اور پہلوؤں کو پیش کر انہوں نے باضابطہ طور پر اپنی شاعری کو محتم کر دیا ہے، یعنی اعلیٰ کر دیا ہے۔ ابھی جیسے ابھی بہت سے شعر پڑھے گئے تھے، نگے کی برچھیاں جو سہ سکے سینہ اسی کا ہے، ہمارا آپ کا جینا نہیں جینا اسی کا ہے، یا اسی طریقے سے جو ان کا بہت ہی لوگوں میں مشہور ہوا اور بیشتر لوگوں کو یہ شعر یاد ہے، کیا یہ بزم میں ہے یا کوتاہ دستی میں ہے محرومی، جو بڑھ کر خود اٹھا لے ہاتھ میں مینا اسی کا ہے۔ یہ دور ایسا چل رہا ہے کہ اگر آپ پیچھے رہ گئے تو پیچھے رہ جائیں گے، اگر آپ اس کو دوڑ کر اور جھپٹ کر لے لیں گے تب وہ چیز حاصل ہوگی۔ شاعر یہی کہتا ہے، یعنی اے انسان، ہمیں ہر منزل کو حاصل کرنے کے لیے خوب تگ و دو کرنا چاہیے، کوشش کرنا چاہیے۔ اس طریقے سے شاد عظیم آبادی کا شاعری کا جب ہم مطالعہ کرتے ہیں تو شاد عظیم آبادی وہ شاعر ہیں جنہوں نے باضابطہ طور پر انسانی اقدار، فلسفیانہ تصورات، سماجی زندگی، دلی کیفیات، حسن و عاشقی، تصوف جیسے تمام مسائل کو اپنی شاعری میں سمیٹ لیا ہے۔ اس لیے یہ کہا جا سکتا ہے کہ شاد عظیم آبادی نو کلاسیکی شاعری میں شعراء میں ایک منفرد پہچان اور شناخت رکھتے ہیں۔ بہت بہت شکریہ سامعین کرام۔
Are you a fresh graduate? Do you want to fulfill your educational aspiration by pursuing higher education in various interdisciplinary and multidisciplinary areas like environmental science, environment and occupational health, philosophy, sustainability science, folklore and culture studies? If yes, the School of Interdisciplinary and Transdisciplinary Studies offers various programs like Master's Program, Graduate Program, Postgraduate Diploma, Certificate and Appreciation Program. The school offers 5 Master's Program. Master of Science, Environmental Science. The eligibility criteria for MSc Environmental Science is Graduate in Science, BTech, BE, Agriculture Graduate, Forestry Graduate and Veterinary Graduate. School also offers Masters in Environment and Occupational Health, Masters in Sustainability Science, Masters in Philosophy and Masters in Folklore and Culture Studies. The eligibility criteria for these 4 Masters program is graduate in any discipline. The school also offers various socially relevant and need-based PG Diploma program like PG Diploma in Sustainability Science, PG Diploma in Environment and Occupational Health, PG Diploma in Folklore and Culture Studies, and PG Diploma in Migration and Diaspora Studies.
Welcome in this session of Gyan Darshan, myself Dr. Anuj Purwar from School of Engineering and Technology. Today we will discuss the unit of our one of the program. This is a diploma in Smart City Development and Management. The topic of this session is the Smart Physical Infrastructure, Water Supply, Stormwater and Solid Waste Management. So first we will see some introductory part. What is the infrastructure? Infrastructure systems such as water supply network, stormwater and solid waste management constitute the foundation of modern societies. Cities have to continuously build, maintain and upgrade physical infrastructure to keep up with the demand of economic development and population growth. With rapid urbanization and increasing demand for services, there is a need for an integrated approach towards infrastructure management. Here the objectives of this unit. Here we will discuss a smart concept for the infrastructure sector, understand the application of Internet of Things in smart infrastructure, describe various sensors used in smart physical infrastructure management, and elaborate on artificial intelligence and its application for the infrastructure sector. So, smart infrastructure. Smart infrastructure is the basic foundation of a smart city. In smart infrastructure, physical urban infrastructure is integrated with human capital, information and computing technologies like software, server and network. As earlier the utilities were managed using manual schedules, now here in smart infrastructure sensors help in detecting a change in environmental conditions and communicate the same through the network. This can ensure the optimal use of resources and improve performance by bringing in greater efficiency of services within cities. As a result, smart infrastructure has the potential to transform the way we plan and manage infrastructure in cities. Smart infrastructure can operate and control both cyber and physical system. Now, advantages of smart infrastructure: more effective data-driven decision-making system, maximize outputs of existing infrastructure system, optimal utilization of resources based on consumption patterns, anticipating future demands based on real-time data, responsive and resilient to climate change, increase citizen and government engagement. Smart infrastructure design principle: people-centric and inclusive approach that respond to the sustainable development needs of the people. Because end users are citizens, including the older generation and people with disabilities, hence the ease of use and accessibility is foremost importance in designing of smart infrastructure. Climate change, urbanization, globalization is presenting unprecedented challenge to cities, so there is a greater need for small infrastructure to be responsive and resilient to external shocks and unexpected disasters. Smart infrastructure technologies are rapidly evolving. Consequently, small infrastructure should be designed to be flexible, keeping in mind future modifications and enhancement. Smart city infrastructure components may raise risks regarding to cybersecurity and digital data safety related to citizens. So ensuring the privacy and safety of citizens is an important concern and important goal. Now we will proceed to the smart water management. Water management involves managing the diverse water needs of multiple consumers like domestic, commercial, institutional and industrial users. A smart water management system, in addition to managing the water demand, also uses technology to help save water, reduce cost and increase the reliability and transparency of the water distribution system. Here the smart water management system components: smart water management, then here the category water supply system, water body monitoring and smart meter. Under the water supply system there is the leakage reduction, smart metering and water demand assessment. Under the water body monitoring there is water quality monitoring. And one more category is there, water treatment plant. Here the advancement, water treatment and automation and pressure reductions. Role of data in smart water management. The efficiency of water management is based on water data availability and using this data in a suitable and accessible format to make sound decisions. One can collect data for water management using interconnected networks of sensory devices and technology. Now we will move to IoT for smart water management. Before going this, we first introduce the IoT. IoT means Internet of Things. Means here we are talking sensor, some software, some devices, and finally the data processing. So in IoT enabled smart water management system, a wide array of sensors are deployed, throughout the water infrastructure and connected via wireless network. These sensors can measure indicators like volume of water, flow of water, temperature, pressure, vibrations, humidity levels, water levels in tanks, and water demand assessment, etc. So here the few sensors used in the smart water management for the flow measuring, pH sensor for the water quality measuring, smart meters which may find the leakage, and they identify the internal leakage at homes, and pressure sensor. Artificial intelligence in water supply. One of the ways in which artificial intelligence is being used in water management is to monitor and analyze water cycle data. This includes checking water quality, tracking water use, and identifying potential problem in water supply infrastructure. In short, we can say artificial intelligence like we are searching our queries in the Google. So this is the based on the artificial intelligence. Suppose we are using the YouTube. In YouTube, again we frequently search the things. As we can say in Amazon, we frequently search the product. Then this software, this mobile software, learn the our demand, and then basis of this data, what this already accumulated in their search engine, they frequently communicate to us with their new product which is similar to our demand. So in artificial intelligence in water supply, AI algorithms can help the water distribution system to become more efficient. The first step is to use ICT using artificial intelligence algorithm to optimize the number and location of the sensor. The second step is to start building historical data in digital format using SCADA, Network Operations Center, and hydraulic. The third step is use of artificial numerical tools in decision making at the core of planning and operation. Ultimately, here we are discussing the application of artificial intelligence in the smart water distribution system. And basic, when we talking about the artificial intelligence, behind of this artificial intelligence there is the data. Once we feed the data in the machine, as we can say the computer system, and then we can use this data for in decision making. For example, we can say, we can take the example of the video games. In video games, what we do, suppose we are taking the game of chess. So in chess, first we move that first person which is available, and one is a computer. So first person when move the one step, then basis of this step, computer take the decision and make the further step. So another example we can say, today we are talking about the self-driven cars. So here some data is already feed, and in basis of these feed data, this car can be take the decision. So here also we are required the data which is feed in the system or some software system, and basis of this we can utilize the data as per our convenience. So next is a smart storm water management. In similar way, urbanization and land development both are disruptive to the hydrological cycle, as they alter the land cover and promote increased runoff in cities. The conventional design of storm water is based on historical observation of precipitation and land use. The smart storm management approach, on the other hand, is designed to optimize and mitigate the impact of the discharge runoff, both in terms of quantity and quality, using real-time monitoring data. This approach relies heavily on the use of sensor and information technology to make existing stormwater systems more adaptive by embedding them with connectivity and intelligence. Advantages of smart stormwater management: to track and prevent urban flooding, validation of drainage models, use opportunities to optimize full system capacity for both existing and proposed facilities. Restoring pre-development hydrology and base flows by actively modulating release rates based on forecast information. Reducing operation and maintenance cost by issuing alert in real time. Further, the continuous monitoring of stormwater storage can help in maintaining the water quality. Now here we are talking about the real-time control system. Innovative solutions such as real-time control or dynamic stormwater management system can help in managing stormwater management in cities. The goal of an RTC system is to continuously regulate the flow in the various branches of a network based on a real-time information related to system capacity and weather condition, thus reducing the magnitude of outflows during storms and relieving other stresses on the system. The application of RTC requires continuous monitoring, water level, rainfall, weather forecast, etc., control devices, valves, gate, etc., and data communication to actively manage flows and adaptation to change condition. Infiltration rates can be monitored in real time and integrated into the RTC management strategy. Outfitting stormwater infrastructure with sensor and digital control systems introduces new opportunities for efficiency and to optimize existing infrastructure. Now, IoT for flood management. In the case of a flood, the early warning system is most critical for the authorities to predict and minimize the impact of the disaster on the community. The process starts with an ultrasonic sensor measuring the level of water in the river. Next, collected data from the sensor is forwarded to a microcontroller. Then, alarm attached with microcontroller system is sounded to alert the public and authority when there is an upcoming flood. The software components are designed to compute the stream flow and to quantity the spatial distribution of flood risk for each control water set. These solutions combine the constant feed of monitoring data with weather forecast to aid in flood control and increase water quality. Use of artificial intelligence in stormwater management. Using modeling tools and decision support system, the AI-enabled application can control hydrological devices such as pumps, valves, or gates to adjust or control the flow rate of a drainage system. Means we already input the data, and basis of these data may be supervised or unsupervised. Basis of these data, when there is a flood, so automatic pump or valve will be the close. The overall benefit of using AI in stormwater management include an increase in energy efficiency, a decrease in flood risk, and a decrease in cost. Here we are talking similarly what we are using in Alaska in the Amazon, where we just give the voice instruction to that device, and finally they give the desirable result. So here, when sensor is available and data is available, and basis of this sensor and data, this some device recognize the things, and in case of the flood, suppose, so there the gate will be automatically closed. Now, smart water solid waste management. Waste generation is another infrastructure challenges faced by many cities in India and across the world. Solid waste can cause significant health, aesthetic and environmental pollution problem if it is not disposed of appropriately. At present, the urban waste collection process is very complex and require a significant amount of resources. Typical waste management practices include the monitoring, collection, transport, processing, recycling, and finally disposal of the waste. Smart waste management technology can be used to identify waste generation patterns, optimize driver routes and schedules, and thus reduce the operational cost. Smart waste management involves assessing trash bin, fill label measurement, route optimization of the trucks, and also can include provisions which can contribute to the recycling process by easing out the sorting process, which is at present is manually and slow. At present we are sorting the things manually. Suppose there is a certain types of solid waste is available, there is a glass available, there may be the different type of glass available, there may be the plastic, there may be the different type of plastic. Suppose there is metal is available, so metal we can sort it out with the help of the magnet, but we cannot differentiate between the different type of plastic or glass. So here the some smart techniques are available. With the use of these techniques we can fastly sort it out, and precisely and efficiently we can sort it out the recyclable things. Advantages of smart waste management system: reduction in collection cost with smart bin transmitting their real-time fill label information to waste collectors. Means some sensor is available in smart bin. Once they are filled, so this information is proceed to the concerned persons and they will take the decision accordingly. Dynamic routing of waste with optimization of waste collection routes and schedules based on real-time data. Timely and improved cleanliness due to real-time data information, as a result no missed pickups. Understanding of waste generation patterns using data analytics. Direct carbon dioxide reduction, as optimized routes lead to less fuel consumption and utilization of carbon dioxide contributing resources. The IoT for smart waste management. Smart bins and smart trucks are crucial in the route optimization process. Some of the IoT tools used in waste management systems are sensor, cloud systems, wireless technology, and mobile application. Cloud waste analytics can help improve scheduling and optimize truck routes. Using sensor and connectivity, IoT provides a substantial number of ways to manage waste so that it sustainable and environmentally friendly. Here IoT solution is developed in two phases. First is a monitoring phase, where the level of waste inside the compartment are constantly measured, transmitted and stored. The second phase, where the calculation of the collected information is applied to optimize the waste collection routes. IoT for smart bins. Data such as filling label, temperature, what is the temperature inside the bin, humidity and any necessary data can be collected from the sensor. Sensor implementation in the waste bin together with IoT connectivity allow for real-time monitoring. Means one sensor is available inside the bin, and this sensor or this bin is connected some, we can say, some centralized system, processing system, where this data directly comes in that centralized system, and in that centralized system this real-time data comes and based again this data based to the concerned person which goes to the spot where this bin is available, and this bin that is manually, automatically, or whatever the way this is emptied. The level of garbage in the dustbin is detected with the help of a ultrasonic sensor. There are some sensors available. With the help of these sensor, what type of waste is available there, and which is recyclable waste is available, which can be reused, and based on this data, these ultrasonic sensor can detect this type of waste, and this waste can be separated out manually or automatically. Here maybe the bin may be the smart, but system may not be the smart. If every system is smart, then we can say sensor and detect this smart bin is full, then this is emptied smartly, a vehicle will come smartly, and this bin will empty, and vehicles move it's on the destination. The ultrasonic sensor is used to find the height The ultrasonic sensor is used to find the height of garbage filled at different intervals of time. Using the microcontroller platform, the data collected by the ultrasonic sensor is sent to the web page with the help of a Wi-Fi module. A server is used to store data and shows all dustbin labels on the web page. GSM is used to send text messages to the mobile. The text message contains information about the garbage label and location of a particular bin, as already discussed. Then sensor can be mounted inside the garbage bins and connected to a monitoring system that can relay the fill labels of the bins. The amount of waste is determined by the labels of waste collected from the top of the waste bin lid. The temperature sensor and weight sensor along with metal detector are useful for sorting the biodegradable, recyclable and reusable items from the trash cans. Using image recognition and radio frequency identification, some smart bins are also designed to recognize and classify different types of the waste. IoT for route optimization. Using IoT, it is possible to track the location of the waste container, to identify location with the highest demand, and to suggest the shortest route of optimization collection of solid waste. Smart trucks capture and store vehicular data related to the truck's load weight, route status, service completion, and driver activities to provide accurate performance reports and real-time alert. And similar way, we are talking about here the artificial intelligence in waste management. So digitalization and artificial intelligence methods can make a major contribution for improving waste management in the cities. So the role of the artificial intelligence in waste management begins with intelligent garbage bins. For example, some public locations like school, offices, hospital might need immediate or frequent cleaning, so immediately services will be provided to them. Artificial intelligence can also be used for waste sorting, segregation and recycling for having more sustainable solution to the waste generation in the cities. And now thank you.
We wear clothes on a daily basis. But have we ever wondered, where do our clothes come from? Who makes our clothing? And who are the people and the process responsible for the whole manufacturing of our clothing? If you want to know more about this, Indira Gandhi National Open University from the School of Continuing Education has launched a program which is Certificate in Apparel Merchandising. There are many benefits after doing this program. You can become a designer in various fields. It can be fashion designer, apparel designer, costume designer, or even a textile designer. One can also work as a fashion consultant. Research and development is another field which one can take up after doing this certificate course. Going into academics is another perk after doing this program. Getting into fashion houses or fashion studios as an intern is also a very good option post doing the certificate in apparel merchandising. The basic eligibility of the program is 10+2. The duration of the program is 6 months to 2 years. The program is also available for both the admission cycles, which is July and January. The exams are conducted twice a year, in June and December. So what are you waiting for? Join this program and get an enriching knowledge in the field of apparel merchandising. And to do so, you can log on to the official website of the university, www.ignou.ac.in.
Hello viewers, welcome to this program. Today we will discuss on the topic epigenetics and pollution, and this forms a part of the program Postgraduate Diploma in Environmental and Occupational Health and Master of Arts in Environmental and Occupational Health, and in this we have a course MEV004, that is a code on environmental toxicology. So we have an expert with us in the studio today, Dr. Suresh Kumar. Welcome to the studio. And he is a scientist F from the National Cancer Prevention and Research Center, which is an ICMR-based institution based at Noida in New Delhi. Welcome, sir. And I would like you to first introduce the learners and the viewers with the concept of epigenetics, as to what is epigenetics and the mechanisms behind the epigenetics.
Fine. When we talk about epigenetics and pollution, so before going to epigenetics, I want to give us some basic introduction about epigenetics, basic introduction how the epigenetics comes. So we know that our body is made up of cells, and every cell in our body contains a same genome and same set of genes. So we have different organs in our body, and these different organs, they do different functions, but they do bear the same genome. So how this could be achieved? The different organs function differently, having the same genome. So the concept is that certain genes necessary for particular function of organ is only switched on, and rest of the genes are switched off. That means that when we have the 25,000 genes or 30,000 genes in the body, not all the genes are get transcribed, in the sense that not all the genes are going to make a protein. So in spite of having all 30,000 genes in the cells, only particular genes are transcribed. That means that only certain genes are on and rest of the genes are off. So each cell in our body basically express only each cell in a body that express only a small set of genes at any time, particular time and the space. So during development, different cell expresses different set of genes in a precisely regulated fashion. So how this could be achieved? This difference of expression having the same genome, how it could be achieved? So this is what epigenetic comes. So epigenetic comes basically. So coming to this is what I said, that we have different organs in the body. We have a brain, a lung, liver, tissues, many tissues we have. And these all tissues have same cells, but they do different functions. So how we do this? How it could be possible? If you analyze a protein, the differences in protein in the lungs and the liver, you'll always find difference of proteins. So you don't find certain proteins in the liver which is present in lungs. So there are certain proteins which is present in the lungs that will be absent in the liver. That means that those proteins which is absent in the liver doesn't need it for liver function. So this is achieved by switching on and off of the genes. So this is how the epigenetics comes. Before again going to that, I'll come to the basics. So if you see, this is a cell structure. You have plasma membrane. You have inside plasma membrane is enclosed with your Golgi apparatus, and you have a endoplasmic reticulum and mitochondria and lysosomes, and you have inside the nucleus. And inside nucleus you will have a genome, and which is made up of DNA. So the DNA is, if coming from from down to up, you see that the DNA is organized into multiple compaction mode, in which the DNA is wrapped around some protein which is called histones. So this DNA is wrapped around histones, and this histones again organized into solenoid model, and this model is being formed into a chromosome structure. So this is a multi-order structure which is achieved by coil way of packaging into chromosome structure. So this is how DNA is packaged in this cells. So if you see that this is what the developmental marking of establishment of epigenetics. If you see that when the zygote is formed, when the zygote is formed, the sperm and oocyte forms. It forms a zygote, and it becomes 2-cell stage, 4-cell stage, and 8-cell stage. So at these stages, they are basically we call as a totipotent cells. These cells are can form to any organs. But once this cell stage forms into 8-cell stage and its 16-cell stage, this totipotency is lost. So when the totipotency is lost, and further cells like 16 cells and 32 cells, they have a different marking in each cells. So each cells get differentiated to form particular organ. This is what we call as a differentiation. Like you have ectoderm, mesoderm, endoderm. So this is what marking that happens at this stage. So how this marking? That is marking by some methyl group. This is what we call DNA methylation. So there is a methyl group is added in your DNA that actually acts as a signature for a particular cell type. So this is done by DNA methyl transferases, which is we call as a DNMT1 and DNMT3A, 3B. So DNMT3A, 3B are de novo methyl transferases that acts at the embryonic pattern when it sets up a DNA methylation pattern. And DNA methyl transferase 1, actually it is called maintenance methylation. So these enzymes are very important in establishing methylation as well maintenance of methylation. And this methyl group in the DNA actually is different from cell type to cell type. So you have same DNA, but you have different methyl marks. So that actually defines your gene expression pattern. So that's how different organs have different functions. So that's how the cells of different organs, they give different protein molecules for its function. So this is what central dogma. So DNA is transcribed into RNA, RNA transcribed into this RNA get processed, and it get translated in the cytoplasm into protein. So this central dogma is that which to say that this DNA, all the DNA is not going to get transcribed. Okay. So how it is not transcribed? So at every level you have control, transcriptional control, once before DNA transcribed into RNA, there is a transcriptional control. We call it as a pre-transcriptional control. This is where epigenetics plays a role. And once the RNA transcript is formed, then it get to processing control. That mRNA become translocate to cytoplasm for translation. So then again, at every level you have a controls. So after this many controls, the protein has to be produced. So you have multiple level of controls for your gene expression. So now what we are seeing is that epigenetics plays at the pre-transcriptional control level. That we will be seeing it. This is a simple example which I have given to understand that nobody likes a baby to have the hair growth in their body and with the mustaches. So baby has to be like a baby, right? This is how everybody likes. But the question comes that the baby has the same gene what I have. The same, but why this baby is not having mustache? When the baby bonds or when the baby stays in its childhood, it is not having mustaches. Why? But the genes are there. So why I am putting this example is that there is a concept called spatial and temporal gene expression. Spatial is space, temporal is time. Certain gene has to be expressed only at particular space, that is a spatial. Certain genes has to be expressed only at particular time, that is temporal gene expression. So the gene responsible for secondary sexual character, for mustaches and all, has to be expressed once a child attains the age of 13 or 14 years. That is controlled by endocrine systems. Sexual hormone started producing its hormone, then it starts showing its secondary sexual characters. So these genes are shut off when they are in childhood. This shut off mechanism by DNA methylation. DNA methylation at particular genes actually suppresses gene expression in the sense that genes are silenced at particular time and space. Now coming to epigenetics. What is epigenetics? The term epigenetics is coined by C.H. Waddington in 1939. So he showed that the causal interaction between genes and their products which bring the phenotype into being. So this is a little bit of confusing definition, and now what is a reason to understand that epigenetic is a study of heritable changes in gene expression that occur without altering change in the order of DNA sequence. So it doesn't, the change in gene expression is not due to order of DNA sequence. It actually alters DNA sequence by modifications, DNA methylation. The mark is put it on the sequence. So that way it controls the gene expression. So epigenetic changes, epigenetic is a study of gene expression that occur without altering the DNA sequence. So what are the mechanisms, epigenetic mechanisms? So epigenetic mechanisms are DNA methylation, which I have given you. So epigenetic mechanism, DNA methylation and histone modulations and non-coding RNA. So epigenetic mechanism is not only by DNA methylation. So basically here I have written you that this particular cytosine molecule get methylated at particular fifth carbon of cytosine. This become methyl cytosine. This methyl cytosine actually suppresses gene expression by inviting some suppressive complex. That I will come to later on. And also histone modifications. We saw that the DNA is wrapped around the histone molecules, and these histone molecules are organized into solenoid structure. When you have the modification in the histones, so as you know that all the proteins undergo post-translational modifications. When they go post-translational modifications, particularly histones, they do methylation modification as well as acetylation modifications. So that modifications alters the gene expression. Third one is non-coding RNA. This non-coding RNA is basically, this doesn't do any function, they doesn't code for any protein, but they do function by eliciting some gene expression by controlling gene expression. So this all helps in gene silencing or expression of gene expression. So I'm talking about the DNA methylation now. When we take that DNA methylation, generally a classical example is that 4 nucleotides that make up a DNA: adenine, guanine, thymine, cytosine. So this adenine, guanine, thymine, cytosine, the important part for the methylation concern is that cytosine. The cytosine molecule, the fifth carbon portion, the methyl group is added here. this methyl group. So this is called 5-methyl cytosine, which we call. So now this is what the 5 bases what we have known, but the recent technologies have come up that not of 5 bases in the genome. We do have 8 different bases in the genome. That is 5-hydroxymethyl cytosine and 5-formyl cytosine and 5-carboxyl cytosine. But of course, this all these 3 more products actually they are modulations of methylated cytosines. They get oxidized into different formats. So that is what hydroxyl methyl cytosine and formyl methyl cytosine, carboxyl cytosine. These are all modulations of 5-methyl cytosine that happens in the genome. So this is what methylation, methyl group, addition of methyl group in the cytosine. This is what called methylation process. This methylation process that have a role in gene expression. If you see, this is a classical gene. This is something gene structure. When you have a gene structure, you have this is what transcription start site. You have that arrow here, and this is what we call gene. The gene is altered by yellow colored exon and blue colored introns. So exons, introns, it's been alternated. So this is a gene. And this gene control region is that upstream of the gene. This is called promoter region. This is called promoter region. This is the promoter is a place where the control of gene expression happens. So whatever we saw that methylation mark in the cytosine, that happens in this particular promoter region. If this particular promoter has CpG sites, cytosines followed by guanines, which we call CpG sites, multiple CpG sites, they are prone to, it's not a prone, rather it is possible for DNA methylation based modifications. So if the promoter has CpGs, they are highly possible that this particular gene is regulated by methylation process. So this particular promoter will have multiple CpGs and they'll have some transcription regulation, either suppression or expression based on that. So CpG, what is a CpG rich regions? What is CpG island? Island, what we say that island is something which is something isolated in the genome, C of the genome. But of course, this is we call as a CpG rich regions, which are if it is a more than 50% of CG regions in the promoter region, we call as a CpG rich regions. So this to study a CpG rich regions, it has to have more than 50% of CG's regions in the promoter. So the CpG's rich region associated in the promoter region, then most of the genes have control over gene expression through epigenetic modulations. So how this has been controlled? So as we say that this is what CpG island. CpG island, this is CG sequences in the promoter region. And this arrow is basically transcription start site. This CGs, it is not necessarily has to stay only in the promoter region. It can be a promoter region plus it goes up to first exon of any gene. So the methylation in this actually alters gene expression. So when this lollipops which I put, it is not methylated. So your mRNA is produced. This mRNA may produce a protein. Now when these lollipops are methylated, when the CGs, when the cytosines are methylated, then transcriptional silencing happens. This CGs actually invites some of the repressor complexes to bind with that so that RNA polymerase cannot come and sit, so that gene silencing happens. So this is how cytosine methylation mediated gene silencing happens. Here you see that this is a repressor complexes in CGs and these are transcription factors. When there is no any CpGs methylated, when there is no CpG methylation, the transcription factor come and sit here and the transcription can go on. But when this is methylation, this is filled lollipops, this is when there is a methylation is there, the transcription factor cannot come. So this actually interrupt the binding of RNA polymerase and transcription factor, and it leads to gene silencing. So this is simple DNA methylation mediated gene silencing method. So role of DNA methylation.
So now that you have explained about the epigenetics and also one of the mechanisms of epigenetics, that is the DNA methylation, and you also explained about the 5 bases and also some more in the nucleotide. So is there any clinical relevance of the DNA methylation?
Okay, DNA relevance to methylation. Okay, This methylation basically happens by DNA methyl transferases. Once the cells are replicated, like in post-replication, it copies the mother's DNA, how the the template DNA is methylated. According to the template DNA methylation, the daughter DNA, once replication is finished, the daughter DNA also get methylated based on the mother's pattern. So that is done by DNA methyl transferases. The DNA methyl transferases does this by using S-adenosyl methionine. It is basically methyl donor. So it takes a methyl group from this S-adenosyl methionine for this process. How this SAM has been come up? So SAM actually, for synthesis of biosynthesis of SAM, you need externally supplemented or in food items you have to take. The one of thing is folic acid. So during trimester in the pregnancy, the pregnant mothers were given folic acid and iron supplementation for a particular time. This folic acid is given to ensure that the methylation, the methyl group has to be established in embryo, the developing embryo, so that the normal development has to happen. So this has been proved when there is a deficiency in methyl groups in the mouse models they develop. When there is a methylation deficiency, they found that there is a neural tube defects.
Oh, okay.
So it is to ensure that normal development, methylation is very much necessary. So they have given folic acid. So folic acid is basically substrate for, providing the methyl donor to the S-adenosyl methionine formation. So that has a clinical relevance. It's a simple clinical relevance which I have given.
Right.
So roles of DNA methylation. So as I said that the DNA methylation is good and it is needed for the health as a body. So it has a role in transcription silencing. I said that certain genes has to be active, certain genes has to be off. So for that it has a role. And also it protects the genome from transposition. Some of the genes called transposons, they has to be protected. The transposons should not be get activated. Once if it is activated, it forms as jumping genes and it forms the genomic instability. So the transposons basically has been suppressed by methylation process. X inactivation, unsexed domain. X inactivation, the females have 2 X chromosomes. One may argue that the X specific genes will be more in the females, but the methylation does justification. One of the X is inactivated in the females, so that the X inactivation happens due to DNA methylation. And also it helps in, it also play a role in sex determination. And the aquatic animals, some of the temperature based sex determination happens. So temperature actually alters the epigenetic patterns that elicits some of the sexual hormones to be produced, and change of sex or different sex determination happen due to methylation. So these are the roles of DNA methylation. So with that I'll...
Thank you, Dr. Suresh. So today, my dear learners, we have understood the concept of epigenetics, and then you also went on to describe the processes that are involved. We will be continuing our next session. So one of the things that you explained in the mechanism was the DNA methylation. And then you also explained the relevance, like for example, the clinical relevance. One of the examples were that of the folic acid that were given to the women who were in their gestational state, and how that is also very important for preventing the neural defects among the newly born children. So now we understand why this study is also very important, and it is of very good societal and scientific relevance. And in the next session we will discuss about the next, maybe the histone modulation and some more to be understood, and also whether pollution, environmental pollution, can also have effects on the DNA, on some of the changes that we are observing, maybe even in genetics. Does that have a role to play? So because these days we are seeing a lot of pollution related ailments, and these things can also be interrelated. So thank you for this session.
Thanks.
And thank you, learners. I hope you have understood some of these concepts, and which will be useful in the course related to environmental toxicology. Thank you for your patient listening.
Thank you.
Do you wish to be part of a television studio like this or any other media system? Or already part of any media system, want to enhance a new set of skills? Welcome to School of Journalism and New Media Studies. Here in the School of Journalism and New Media Studies, we are offering program for all set of people to learn and enhance their media skills. The vision of School of Journalism and New Media Studies is to generate a new set of professionals in the area of media and communication studies with understanding about role of media in the national development and global understanding. We offer innovative programs in the field of journalism and mass communication that sets the benchmark of high quality learning and teaching process at the national level. We offer these programs across the country with the help of our regional network as well as study center. Same manner, many of our programs are offered through online mode with a sophisticated learning management system. SOGNMS offers programs from PhD in journalism and mass communication till certificate level. We have masters in journalism and mass communication. The eligibility is bachelor's degree, and we are introducing this program in 3 languages: Hindi, Tamil and English. Students from national and international students can join either through online mode as well as in the distance mode. We have 4 postgraduate diploma programs, specialized areas in the field of development communication, digital media, electronic media and journalism and mass communication program. And all these 4 programs are available in distance mode. 2 programs are available in online mode, development communication and digital media. And we have specialized certificate program in community radio. IGNOU employs various media and communication tools for better teaching and learning process. School of Journalism and New Media Studies utilizes this media system for delivering all our programs. Come and join with us for exploring a new career and enhance your media skills.
Hello viewers and learners, welcome to this session. Today we will discuss on epigenetics and pollution part 2, and this forms a part of the program Postgraduate Diploma in Environmental and Occupational Health and Master of Arts in Environmental and Occupational Health, and this will form a part of the course in the program that has a course code of MEV 004 Environmental Toxicology. Now to discuss this, we have with us in our studio Dr. Suresh Kumar, Scientist F from the National Institute of Cancer Prevention and Research Center Control from the ICMR located in Noida in New Delhi. Welcome to this session and to IGNOU, sir. And in the previous session, sir discussed about the methylation patterns and how we have seen some of the benefits of taking like the folic acid during some of the women who were in their first trimester, and how the iron or the folic acid is important for preventing the neural defects among the babies that were born. So that was some positive outcome of this DNA methylation and the processes involved. And there can also be pollution related problems which can alter this DNA methylation. So I just had one question, sir, that we have seen some of the benefits. So just like this, pollution can have an effect, and if there is some change which is observed, can that be reversible? I mean, can this be reversed back to its original state?
Right. So we can reverse it. May I can go to some more epigenetic mechanism that also have a
Yeah,
role,
yeah, of course.
then I can answer your question.
Definitely.
So we can go for that other mechanisms which we have. Last time we seen a DNA methylation as one of the mechanism that happens. So not a DNA methylation alone that do gene expression regulations. It also takes the help of histone modulations.
That's right.
So I'll come to this histone modulations. So basically histones are a nuclear protein that is wrapped by DNA, and they are found as a nucleosome in the nucleus. So in histones you have H2A, H2B, H3 and H4. These 4 histones they found as a dimers. So these octamers are wrapped by 146 base pair of DNA. So these histones are subject to variety of post-translation modification like any other proteins. The proteins, once it has been translated, they do undergo a post-translational modifications. So the post-translational modifications can be phosphorylation. So most of the kinases present in the cells, they get phosphorylated for its activation. And phosphorylation, acetylation, methylation, and ubiquitination. So ubiquitination generally that is been found in protein degradations. So these post-translational modifications that alters a gene expressions. This is also a epigenetic modification that happens in the histones. We have seen epigenetic modification in the DNA. In the cytosines, the methylation happens. Now we are going to see the modification that happens in histones, particularly in the amino acids. Particular amino acids in this particular histones, they do get modulated by acetylation or methylation. These 2 things which we are interested with our concern with our epigenetic study. So as I seen that again, I'll just recapitulate the things that epigenetic mechanism, DNA methylation, histone modification, and non-coding RNA. These 3 all together, either independently, coordinately, they do gene expression regulations. So as I say that H2A, H2B, H3, H4, they are all found as a dimers. They do have wrapped around by DNA, and this particularly if you see that histone tails here, and particularly H3 histone tails here, this H3 histone basically is amenable for the modifications, either acetylation or methylation. When methylation happens, the particular H3, I mean histone 3, H3 dimethylation, trimethylation in particular amino acid, that is we call H3K4, that is H3K4 trimethylation. That determines the expression. When you have this methylation, this is a transcriptionally active methylation pattern. When this trimethylation become dimethylation, it become repressive. Same way, H3K27 methylation is a repressive mark. So the methylation in K4, trimethylation K4 is basically active form, and H3K27 methylation is the repressive mark. So methylation mark here play a different role based on the trimethylation, dimethylation in the histones, and also acetylation. Acetylation also basically, if you see that acetylation gives negative charge to the DNA. So when the acetyl group is added into the any of the amino acid, particularly lysine again, this amino acid gives attains a negative charge, and net negative charge attained by histones. When histone become negative charge and DNA also negative charge, they repel each other. So acetylation basically induces active gene expression environment. So if the particular histone is acetylated, so that genes which are wrapped around by histones, they become active transcriptally, right? And if you remove the acetylation, it become clumsy, that become tightly coiled. So the euchromatin state become heterochromatin state by removing the acetylation. So this is what you have a different modulations, particularly by acetylation, methylation. This K4, this is a K4 methylation, and K27 methylation is what important in our most of the studies which are related to the environment, pollution, as well cancer biology. So methylation in K27 is being found in most of the repressed state of genes, and methylation in K4H3 region, that is active region of genes. And this is actually, this methylation mark, all other methylation marks are been recognized by set of readers, writers and erasers of methylases, basically histone methylases. So certain histone methylases they reads, the certain methylases they recognizes, certain methylases they erases. So this is a kind of a dynamic part of inclusion and erasure of DNA histone modulations. So organization of epigenome is like that. When this H3K4 trimethylation happens, these are basically transcriptionally accepted region, and when it is H3K27 methylated, it become heterochromatin. This is not transcriptionally active region. This is suppressive state. So this way, DNA methylation and histone modulation acting together, they do transcriptional suppressions. So this is a mechanism which I'm not this. I mean, coming to this part particularly, if you see that this is a gene which is having the second mechanism what I'm seeing here. This is a gene at the promoter region. You have transcription factor binding site is here. When it is methylated, it invades some of the molecule, and this molecule basically MECP2. This is basically suppressive complex, along with some of the histone deacetylases comes here. That actually suppresses gene expression. So this is just how the histones also, histone demethylases have a role in gene silencing, which I am telling. So basically relaxed transcriptionally active DNA is called euchromatin, and more condensed is heterochromatin. Here we see that more condensed, this is what more condensed region, this is what heterochromatin, and this is relaxed is euchromatin. So then I say that acetylation removes the positive charge on the histones, result in repulsion with the negatively charged phosphate groups of DNA. As a consequence, chromatin is transformed into a more relaxed structure that is associated with the greater level of gene transcription. So there are histone deacetylases and histone acetylases. So they do act in different direction, and so condensation can be brought by deacetylating the histones. So that way the regulation of gene expression has been monitored by the histone modifying enzymes. So this is how it happens. So in the normal cells, particular tumor suppressor genes is in a euchromatic state, and when this has been when this particular when this acetyl group is removed in the cancer cells, it becomes heterochromatized. So it becomes a repressive complex. So this is how the histone modulation they have role. So this is another example gene which is in a euchromatic state and become heterochromatic state because of methylation and in conjunction with your histone modulations. So coming to your question, madam. So you asked a question, what is the good effect of I mean, we have seen in the DNA methylation the good effect of this thing. The bad effect, we'll see that whatever this DNA methylases enzymes, it's like we saw that DNA methyl transferase 1 and histone methylases, histone demethylases. This actually, there is a set role to maintain this methylation pattern, and it is inherited from mother cell to new daughter cells. This is exactly same copy is being made. If any alteration in this pattern is again fixed in the alteration, when the alteration happens in this methylation pattern or histone modifications, it is again fixed in the cells. So how we can avoid this? Now what we are now, there are a lot of now the pollution actually causes the defects in methylation machinery. It interacts with the DNA methyl transferase expression, or it intercalates with the DNA strands, the pollutants. So there are a lot of mechanisms which actually alters the enzymatic activity of this methyl group also. So the recent technologies which is coming up, epigenomic studies, they are bringing a lot of evidences, and which we can make that this particular epigenetic alteration can be very well managed by advisories, formulation of advisories, and also lifestyle factors and dietary patterns, how you follow it up. So these things are coming up now, and with a new advent of technologies. Maybe I'll discuss in coming session also, coming about when we talk about the pollution, I'll discuss about those things. So third mechanism is the
So it is possible to be reversed.
Yes,
that's what
this
that was
methylation patterns are reversed, as we saw that when the folic acid, by giving folic acid, we could ensure that neural development is ensured. So same way we can reverse certain epigenetic patterns by management of by diet factors. I'll discuss about that. So the non-coding RNA, basically these RNAs are basically, they don't code for any protein. So they do function in some cellular physiology, but they don't do any functions. So one is that housekeeping RNAs, which we call tRNA, rRNA. These are the one which are normally functioning in translation as well, translation process, protein synthesis process. There is another class of RNAs which are classified based on the size. One is small RNA and long non-coding RNAs. These small RNAs, basically they transcribed into the small transcript and processed into smaller, like 21 bases of RNAs. These are called miRNAs, siRNAs and all. And long non-coding RNA can be more than 200 bases. That is a hot air RNA, telomerase RNA. These are the example for non-coding RNA, which has functional in the cellular physiology, which has a function in cellular physiology. It can act as if you see that the particular long non-coding RNA, they can help in splicing modulations, they can help in, they can regulate the enzyme, they can act as a sponge in the sense that it can regulate the miRNA expression, they can also help in translocating protein to a particular site. So it does a lot of functions. So long non-coding RNAs, small RNAs, they do have epigenetic functions. So the presence or absence of this RNA, they have effect. The presence and absence of this non-coding RNA is mediated by, again, DNA methylation and histone modulations. So now the pollution, coming to the pollution, how it is relevant to the epigenetics. The pollution is defined as any undesirable change in physical, chemical, biological characteristics of air, water and land that may be harmful to our living organisms. So there are different pollutants. There are primary pollutant, secondary pollutant. Primary pollutant, we generally directly face the primary pollutants, and based on the nature, you have solid, liquid, gases, different types of pollutants, and based on decomposition, you can say biodegradable and non-biodegradable. So the epigenome players, basically the proteins which play a role in acetylation, methylation chemicals, they become sensitive to these environmental cues. They become sensitive to these environmental stresses and they get altered. The methylation machineries, the enzymes, the methyl transferases and DNMTs, they get altered because of this particular pollutions. How they get altered, we'll come to see. So there are changes that have important effect in the phenotype of the organisms. They induce the pollutants, they not only alters the phenotype of the cell, it induces changes in DNA methylation, histone modification and also non-coding RNA expression. So generally it alters the epigenetic status of the cells. If you see that this is the chromosome, so you have P arm and Q arm, and this is so this is a telomere at both the ends, and this is a centromere. And these stars which I have written, that this is basically depicting a methylation pattern. So you have, in normal cells, you have hypermethylation and whole genome. Hypermethylation, you have a lot of stars here, that means a lot of methyl cytosine content. Okay, so in the normal cells, you will have a lot of methyl cytosine content, whereas the exposure to pollutants reduces this number of methyl contents, in the sense that the hypermethylation become hypomethylation, right? The number of methylated cytosine reduced here. So number of methyl cytosine reduced here means it is hypomethylation. As you know that there are certain genes are kept off in the normal cells. For example, like oncogenes, proto-oncogenes, like c-MYC. There are other genes called transposons. These transposons we acquire through viral infections. These transposons are kept silent by methylation process. But this pollutions, what it does is that due to hypomethylation, hyper to hypomethylation, the genes which are silenced by methylation, they become active. The c-MYC, it is basically a proto-oncogene. They become active. So these oncogenes basically they helps to cells to proliferate more. Same way that the transposons. Once the transposons get active, they started jumping one place to another place in the genome and creates a genomic instability. So there are multiple jumping genes present in the genome. That means that it's not good for health of the genome. So that way it creates a genomic instability in the cell. So hypermethylation, so here that we see that hypermethylation goes into hypomethylation in the chromosomes. So and also you
So
see that
these environmental changes, like whatever you said now, so they can also be managed in the clinical setup?
yes, this can be managed very much in clinical setup. I'll give you some examples in the coming slides. Maybe some of the examples in the slides. There are a lot of studies have been done. So when the particular pollutants has been exposed to the cells, they do methylation in particular genes, and they have in few studies they have managed it. That I'll tell you.
And also I have some more interesting questions, but according to the thing you can see, like these days, like in olden days we were not exposed to so much of the pollutants, even from the cosmetics or whatever we were using, because we were only using maybe the handmade soaps or the powders or the whatever concoctions that we were getting from the trees or the bark of the trees. But nowadays when you go for a shampoo or a dye product or anything, just flip the label and you find so many of the chemicals in it.
Right. Yes.
So does this also have an effect on?
Yes.
And then still, why are they being sold, and why are people still like wanting to buy them off those?
So this is what the
shelves?
strategy, what you said, that how we can manage clinically or how we can manage it. This is for exposure reduction measures we have to take. It's a personally as well government policies like exposure
Because
reduction.
most of the people will not be knowing this.
Right.
You want to flaunt nice hair.
Right.
You just go for the product.
Exactly.
So they are not able to explain.
Right.
And I often go and I ask them that there are a list of the chemicals and still you're asking me to buy. So where is the logic in this?
Right.
So does that also have an effect on the
Exactly.
DNA? On
It has an effect, yes. Okay. All the things which is, whatever the abnormal things, whatever we have against nature, that all have, that alters the epigenetic. And whenever using, there is a short-term usage creates some short-term usage creates a hypermethylation. Some of the chemicals, when you do chronically, they have a different methylation patterns. That we can see that. So as we see here in this particular slide, we have a hypomethylation, for hypermethylation to hypomethylation in whole genome that happens. Yes, this is what the slide. So here, but if you see the regional hypermethylation happens, the regional hypermethylation happens here. Certain genes are heavily methylated, whereas this is not so. Only particular region is hypermethylated. So genome hypomethylation and regional hypermethylation that happens in the cancers, particularly when it is exposed to certain pollutants. So there is a regional hypomethylation in the normal, and whereas the cancers, it is regional hypermethylation in the cancers. So we know that, as I said, that I'll come to your point by toxins. I'm just telling about toxins. So we see that arsenic is heavily contaminated. I mean, arsenic toxin we do find nowadays in the water, contaminated drinking waters. So the arsenic, basically even Gangetic Plain, the arsenic contamination has been found. The arsenic, what it does is that when we ingest arsenic contaminated water, they get detoxified. So when this arsenic during the process of detoxification, they need this S-adenosyl methionine again for biotransformation for its detoxification. So what it does, it tries to take up all the SAM, S-adenosyl methionine, and it gets transformed into multiple forms. When SAM is been taken out from the cellular pool, the cell become deficient of SAM pool, that methyl donor pool is been deficient. So what happens is obviously the normal cellular process which you need to have hypermethylation, the genomic hypermethylation, that become hypomethylated. So this way arsenic extracts all the pool and it induces hypomethylation in the cells. Same way cadmium. Cadmium shorter exposure, it induces particular gene, suppresses a particular gene expression like hydroxysteroid beta hydrogenases 2. And same way cadmium, a longer exposure that elicits a hypermethylation of P15, P16 genes. This P15, P16 genes are cell cycle regulated genes. These are basically the genes which acts on the initiation of carcinogenesis process. When this P15, P16 are suppressed, then the cells are prone for further epigenetic insults that may lead to carcinogenesis process.
So this may also be one of the reasons that these days we are finding a lot of the carcinogenic and cancer cases.
Right.
Because the water is totally contaminated and you find a lot of heavy metals, industrial pollutants.
And heavy metals, not only the abstracts as SAM, it also induces reactive oxygen species once it's get into the cells. So it generates a ROS. When it has a reactive oxygen species enters in the cells, I mean not enters, it's created in the cells. So it induces a DNA damage. So when they induce a DNA damage, that means it also induces a mutation in the cells. So mutation and methylation and coordinately helps in carcinogenesis process. So it has a lot of effects, this metal exposures. So mercury exposure also the same way, nickel exposure is the same way. So there is another pollutant called persistent organic pollutant, which is something stays in the earth for more time. It is more than 60 days, it is not degradable. So these are basically comes from pesticide storage or agriculture fields, everything it comes back. And when it comes to the animals, when animals eat some of the grains and grasses, these animals, when human consumes, all the pesticide comes to the human body. So the human body accumulates this pesticide, which is not degradable. So they also create a modulations in histones and expression and this thing. So there are around 19 chemicals that have been called as POPs, but now government has been telling that it's more than 200 things comes into the as a POP. So we don't have list. A lot of lists are which can create a epigenetic changes in that. So for your question, so one of this is one of the study which they have taken that the bisphenol A, which is bisphenol A present in the plastic bottles, water bottles, even feeding bottles particularly, yes. Bisphenol A has been noticed in this water bottles and feeding bottles, and they have done an experiment with the animal models in which that bisphenol A ingested animals generally normally they develop a brown color coatings. When bisphenol A consumed rats, I mean mouse models, they formed yellow color coats. So when they found out why this is happening, they found out the color gene which is called agouti gene, that the upstream of agouti gene has some transposons, they get activated because of bisphenol A. So because of hypomethylation of transposons, they get activated, and that actually changes the color code of your this thing. So for your questions, when we have this many bisphenol A has been consumed in the same mouse model study, they supplemented this mouse with folic acid. This mouse has been supplemented with the folic acid, and this folic acid supplementations nullify the effect of BPA. Okay. Right. So the mouse model which has been a yellow color, it become brown color back, or the brown color is not transformed to yellow color. So they found that supplementation of folic acid has a role in beneficiary effect in altering DNA methylation. So like many of these metals have a different, so the arsenic has global methylation, they have a regional methylation P16, there is a death associated protein kinase methylation. So many genes are methylated by arsenic and many genes are hypomethylated. So many environmental chemicals, they do alteration in the genes. These are the list, there's a growing list that alters DNA methylation as well as histone modulations. So also miRNA also, like aluminum, they do induce some miRNAs which are basically not good for health, and also mercury also they does some hypomethylation. So every pollutant, they do alters methylation pattern of normal methylation pattern of the cells, and it indirectly helps in developing the same. So in summary, I'll say that so alternation in DNA methylation provides clue for identifying the pollutant based alternation methylation, and also physiological consequent that we can identify by assessing the DNA methylation. And also this alteration, they do alter the DNA methylation and many of these histone modulations, and there are P16, P15 methylation happens in the cells, which shows that these are the cell cycle control. This itself shows that when there is a pollutant affects this particular genes, we have to be a little bit careful that these are the genes which are cell cycle regulatory genes. When these are shut off, the cells keep going on in their cellular progression. So we have to take care of this thing. And as we say that this can be reversed by adopting lifestyle dietary factors. So one more example which I can give is that since we are living in a heavily contaminated air polluted areas like in Delhi, we have that the particulate matters, 10 microns and 2.5 micron particulate matters. The 2.5 particulate matters create a lot of epigenetic defects. It induces immune deficiency. It alters a lot of epigenetic machineries. So P25 and this 2.5 micron particulate matter. One of the study from Columbia University in 2013, I hope. What they did is that they have experimentally exposed the human with the particulate matter for 10 days. Within 3 days, they do find changes, epigenetic changes. In 3 days, they found epigenetic changes. So they go through the 10 days of exposure of this 2.5 micron particulate matter. So the humans exposed in a control environment with this, then they have given supplements. They gave vitamin B. So when the vitamin B was given, the epigenetic alteration that is being induced by pollution has been reversed. So the important observation is that we have to reduce our exposure reduction strategies. We have to develop exposure reduction strategies. We have to adopt a lifestyle in which that it is necessary to keep our health important by consuming healthy diets which has all these vitamins, folic acid supplements, vitamin B, C, folic acids, B12. They all help in ensuring your normal methylation pattern is setting up in the normal status in the cells. So I think with that, if you have any questions, you can. Thank you.
Thank you, sir, for explaining like the epigenetics. And in this session, we actually discussed more about the effects of environmental pollution on the DNA, and you gave a lot of examples about the heavy metals in particularly the arsenic, then cadmium, and also mercury, and also another important thing that was bisphenol A, which is also found in many of the plastic bottles, and so that is why people are now turning to even the metal type of the bottles. But again, though the silicone nipples that are found on the bottles also, there are several issues. So there are environmental pollutants at every step. There is so much to be discussed. But as you say, maybe there could be the problems can be diagnosed, and we need to come up with eco-friendly solutions. And one thing is, yes, these dietary patterns and taking of these B complex and folic acid, which of course under the supervision of medical practitioner, we need to take them in the right dose, and that will definitely have a very good effect on our health and the living systems. So thank you, sir, for explaining all the concepts. And my dear learners, I hope you have had a good understanding of this subject. Thank you for your patient listening. Thank you.
Thank you.